If you're looking for a brief introduction to DNA and RNA, what they're for and how they go about doing it, then welcome to Genetimix. My name is Tim, and this channel is dedicated to genetics and a mix of science communication and critical thinking. DNA is the blueprint for an organism, providing a template for the RNA and proteins that do the actual work in each cell. RNA is very similar to DNA, but is a bit simpler and less stable. RNA stands for ribonucleic acid. DNA has an extra deoxy on the front to make deoxyribonucleic acid. The deoxy actually means that it's missing one oxygen compared to RNA on its ribose sugar, which forms part of the DNA and RNA backbone. The reason why RNA is considered simpler is because it's usually found in a single strand, whereas DNA spends most of its time as the classic double helix structure most of us are familiar with. DNA on the chromosomes is organized into genes, each of which can produce one or more products, either RNA or protein. I'll be talking much more about genes in the next video, so subscribe if you don't want to miss that. The way the system works is that DNA is transcribed to RNA. In some cases, the RNA is the final product. However, in most cases, the RNA is just a temporary intermediate, which is then translated into a protein sequence. In eukaryotic organisms, which includes most of the multicellular organisms like us, the transcription happens in the nucleus, which is where all the DNA is kept, and the RNA then moves outside of the nucleus to small cellular structures called ribosomes, which are attached to membranes in the cytoplasm. It's here that the protein translation and synthesis takes place. Most of the proteins produced are enzymes, which is the term for a biological catalyst. So if you remember your basic chemistry, this type of protein vastly speeds up chemical reactions in the cell. Some proteins are not enzymes. Cell surface proteins are one example here. These live on the surface of the cell, embedded in the outer cell membrane, and can help with communication and signaling. RNA and DNA both have a genetic code, a simplified representation of the sequence of the nucleic acids in each molecule. DNA involves 4 nucleotide building blocks, called adenine, guanine, thymine, and cytosine, which are often abbreviated to A, G, T, and C, the 4 letters, also called bases, of the genetic code. It's worth reiterating, though, while we can represent these as letters, in the background, they are still molecules which react chemically with one another. These simplified representations are actually much more complex in reality. Another key feature of DNA is its double-stranded nature. This is where the story starts to get really interesting. The nucleic acids on either strand form pairs. The C will always pair with G, with A always pairing with T. This pairing, or base pairs, is what gives DNA its robust nature as a template. In order to copy DNA, all you need to do is unzip the strands, and then each strand will form a template for the other. By matching the complementary nucleotide when adding new nucleotides, the sequence can be preserved, and this is exactly what happens when DNA is copied. We'll be coming back to copying shortly. The RNA sequence is very similar, but RNA replaced the thymine nucleotide with uracil, so the RNA alphabet is slightly different to the DNA one. Proteins also have an alphabet, although theirs is more complex, consisting of a pool of 22 amino acids, which are the building blocks of proteins. So, how do we get from an alphabet of 4 letters in DNA and RNA to an alphabet of 22 letters in proteins? This is why this step is called translation. Each amino acid has a triplet code, so a sequence of 3 nucleotides is needed to identify the correct amino acid. Because there are 4 letters in the nucleotide alphabet, there are 64 possible combinations of 3 of these letters, which is more than enough for the 22 amino acids, with most amino acids having 2 or more triplets that will match. For example, this one can be G, C, and then any of U, C, A, or G to give you the alanine amino acid. This gives the genetic code some redundancy and explains why some mutations, changes to the sequence, even in vitally important genes, will be neutral and have no effect on the resulting protein. DNA copying typically takes place during cell division when a cell splits into two copies. There are two types of cell division in eukaryotes. The more common one is called mitosis, and it happens all the time. Mitosis is how you went from a single fertilized egg cell to the 30 trillion odd cells you have today. And each cell has about 2 meters of DNA in it, over 3 billion bases or letters. That's a lot of DNA that needs copying. The other form of cell division is called meiosis, which is the one that produces egg and sperm cells, each with only half the usual amount of DNA. DNA is organized into 23 pairs of chromosomes in humans, so 46 in total. In other words, your DNA is split into 46 chunks. Humans are diploid organisms, which means we have 2 copies of most of our chromosomes. One of which came from our mother via the egg cell, and one from our father via the sperm cell. The exception to the 2 copies of each chromosome rule is found in chromosomes X and Y. You either have 2 X chromosomes, making you biologically female, or 1 X and 1 Y, biologically male. At least, this is the usual situation if all has gone perfectly. There are a number of genetic conditions that are known to be caused by unusual copy numbers of chromosomes. Trisomy 21 is the most well-known example, also known as Down syndrome. As the word trisomy infers, it's caused by having 3 copies of chromosome 21 instead of the usual 2. There are also conditions linked to the X and Y chromosomes affecting sex determination, and I'll be covering some of these in a future video. To round out this topic, we're going to have a look at why DNA is more stable than RNA. The first has already been mentioned, which is the lack of the hydroxyl group that gives DNA its deoxy prefix. This makes DNA less prone to chemical interactions and breakdown. The double-stranded nature of DNA also helps to make it more stable. The nucleotides that pair with each other across the strands are connected together via hydrogen bonds, which you may recall from chemistry classes are weaker than covalent bonds, but still provide significant additional stability. You can find hydrogen bonds in water, which account for its higher than expected boiling point, and they're also to be found in protein molecules, helping them to provide and maintain their 3-dimensional structure. I've mentioned nucleotides a couple of times. Each nucleotide consists of 3 components: a ribose or deoxyribose sugar, which is a simple 5-carbon ring-shaped sugar molecule, a phosphate group, which is a phosphorus atom surrounded by oxygen atoms, and one of 4 nuclear bases, which provides the letter designation. The backbone of DNA and RNA is formed of strong phosphodiester bonds between the phosphate of one nucleotide and the ribose of the next in the chain. The carbon atoms in the sugars are labelled 1 to 5, and the phosphate groups bind to sugars 3 and 5, which is why DNA is spelled from the 5' to the 3' end. You frequently see the usage of 5' and 3' when discussing DNA analysis. The phosphodiester bonds have a negative charge, which also helps to maintain the DNA structure. Under the right conditions, DNA can be preserved for a long time. DNA will last longer if preserved in low temperature, low humidity, dark, anaerobic or oxygen-free, and slightly alkaline conditions. The mineral hydroxyapatite, which is the primary component of bones and teeth, chemically binds to DNA and protects it from degradation, which is what allows us to collect ancient DNA from old bone samples. As you may be aware, DNA testing has become the gold standard in forensics. It is also the gold standard in biology. DNA evidence is practically impossible to argue with. We'll be diving into the details and some examples in future episodes. As mentioned earlier, a longer discussion of genes will be up next, during which we'll build on our understanding of DNA. And that wraps us up for today. If you like this kind of content, please drop a like on the video. If you'd like to see more, please consider subscribing to the channel. If there's anything else you'd like to see covered or have any questions or other points to make, then please drop a comment below. If you'd like to support the channel more directly, then please join our Patreon. There's a link in the description below. And remember, science is amazing. Thank you for joining me, and I'll see you again soon.